So welcome everybody. Today we're going to be talking about the state of Squamata. So for those of you who don't know what squamates are, their living representatives are modern lizards and snakes. They are also lepidosaurs, which means that they're also related to the tuatara, although the tuatara is an outgroup to squamates, so it's not actually included within squamates. And yeah, so I'm going to be the moderator for today's session. My name is Meghan Forcellati. I'm a first-year PhD student at the American Museum of Natural History of New York. And today, as panelists, we have Amelia, Tiago, Mike, and David. And all of them bring incredible expertise. So I'm very excited to have all of them on board with this panel. They all do a very broad range of research across squamate evolution in general. And so I wanted to begin by asking you all if you mind introducing yourselves with your name, your pronouns if you're comfortable sharing, your institution, your position, what you study in 1 to 2 sentences, and your favorite squamate and why. So Tiago, you're the first on my screen. Would you like to start?
Sure, I will start. Hello, everyone. It's really a pleasure to be here. And just to briefly introduce myself, I'm Tiago. I'm a new arrived faculty at Princeton University. I work on the phylogenetics and macroevolution of squamates using the extant and fossil record of squamates, and combining the morphological data with genomic data to understand long-term evolution processes. I have my favorite squamate is, I guess, dibamids, just because they're terribly difficult to place in the squametry of life, so they're just crazy weird and mysterious. But other than that, all are equally favorites to me. Did I miss something?
No, you got it all. Thank you so much.
All right.
All right. And next up, Michael, you're next on my screen.
Sure. My name is Mike Caldwell. I'm a professor at the University of Alberta. I've been here for the last 23 years, and I'm now full professor. I did my graduate work at McGill. Maybe that's an interesting piece of trivia with Bob Carroll. And if there are any, if there are any questions about Tiago's training, well, I'm responsible for that too. So sorry about that, Tiago. But in any case, favorite squamate. Let me think. Good heavens. People would probably think it's snakes, so let's just go with snakes, broadly speaking. But as we'll see through, I think, the rest of the conversation on this topic, that I've kind of worked on just about all of the major groups in some form or another. Maybe nothing published in particular on amphisbaenians, but I have a few ideas. They could rate as my other weird favorite group. Did I hit the high points, Meghan?
Yeah, you did.
Okay, good.
Awesome. And next we have David.
Hi, I'm David. I'm a 6th-year PhD candidate at the University of Texas at Austin in Melissa Kemp's lab. My pronouns are he, him, and what I study broadly anatomy of squamate reptiles. I started specializing on anguid lizards early on in my undergrad, and I've broadened out since then. And I also study Pleistocene reptiles and amphibians, and trying to understand the responses to environmental changes through time. And my favorite squamate is probably the Texas horned lizard, Phrynosoma cornutum, because it's a really interesting lizard with really interesting anatomy, and it's from my home state of Texas, and it has really interesting defensive displays where it can shoot blood out of its eyes, and I think that's really cool.
Awesome. And last but not least, we have Amelia.
Hi, my name is Amelia Zietlow, pronouns she, her. I am also a student at the Richard Gilder School at the American Museum of Natural History in my 4th year. I work on mosasaur morphology primarily. And My favorite squamate, perhaps surprising nobody, is Tylosaurus, because they're big and cool, and I like them a lot.
Great. Thank you all so much. These were such cool answers. Now, I wanted to begin by asking, to someone who's brand new to paleontology and who might not necessarily be sold on studying squamates, what would you say to that person about why this group is interesting and important to study? And I guess we can go in the same order that we started with. So Tiago, if you wanted to answer first.
Okay. All right. So yeah, I guess in general, probably each person will have different answers to this. So I'm very curious also to see each other's answers, what they consider the most interesting aspects to studying squamate evolution and squamate paleontology, which is the core of this. In my particular case, at least what attracted really me the most to study squamate paleontology is not so much about the paleontology component itself only, but just that squamates in general, they really offer an opportunity to study a system that offers a large array of extant species from which we can collect a lot of anatomical data and molecular data, and also a relatively rich fossil record that allows us to test those questions from multiple fronts. So I am first and foremost someone who's deeply interested in evolutionary biology across deep time scales, and see the fossils as an amazing window into a direct window into some of these processes across deep time. And so paleontology is simply, and quite importantly, one of the unique opportunities to understand better those ideas and test those hypotheses from an extra angle, which you cannot do with some other organisms. For instance, with crocodiles that have a much vast diversity in the distant past compared to today. With extant crocodiles, you can study, get some knowledge about their molecular diversity or anatomical, but most of it is only in the fossil record. And squamates have a vast one, just like we also have for mammals. In fact, we have twice as many species of squamates than mammals. So we can really learn a lot from them under multiple angles. So that's really one of the things that attract me so much into using the squamate fossil record to answer various kinds of evolutionary questions.
That's such a cool answer. So, Tiago, for people in the audience that might not know, roughly how many species of squamates are there?
Yeah, absolutely. So we have just over 11,000 species. And importantly, just a decade ago, we thought this was probably around 8,000 species or so. So the number of species being described and published every year is much higher compared to any other vertebrate group. Some years we have 150 species being described every year. So we are still, we still have a very basic knowledge of extant squamate diversity, to be honest. So it's not just the fossil record that we need to improve a lot, it's actually knowledge of the extant record as well. Although we're doing a really good job on making a lot of those going extinct really soon. At the same time, there is a lot of undiscovered encrypted diversity that still waits to be found by researchers.
Yeah, so that's a really great answer. Thank you. And thank you for, I know I put you on the spot a little bit, but that was also really great. So for those in the audience that were listening to that, what Tiago basically just said is that squamates are one of the most speciose vertebrate lineages. They have some of the most species out there, and there's so much undescribed. So that's also a really great answer. So thank you. All right.
Mm-hmm.
And next up, we have Mike Caldwell.
Sure. Tiago pretty much covered all the points I would have tried to make, Meghan. But I'll just add to that by reinforcing everything that he said. I completely agree. And, to give a little bit of history, which might also be a bit of a motivation for other people, I didn't enter the business of vertebrate paleontology thinking I would work on squamates. It all happened by accident. I went to Bob Carroll's lab thinking I would be a synapsid guy because Bob had published a lot on synapsids. He had completely different ideas, and I ended up doing something on limb evolution in secondarily aquatic vertebrates, well, reptiles. And out of that, I got introduced to fossil squamates when I was banging around in collections looking at limb patterning. Because one of the great things about the marine fossil record, as many of you know, is it will preserve quite often in good and complete detail articulated limbs and skeletons and all that sort of stuff, whereas terrestrial environments aren't always quite as productive. Taking that over to the squamate side of the story, so we've got a good fossil record, and I started out with mosasaurs, much like Amelia right now. Taking that over to the extant record, what I think is really riveting is not just the 11,000 extant species of crown squamates, but the whole problem we have in reconciling interrelationships as we think we understand them from the crown with what we get out of the fossil record. And then on top of that complexity, which is a challenge as an evolutionary biologist, because you need to be an expert on both living squamates and what the fossil record has to say about members of Squamata that might nest within the crown, or more intriguingly yet, might actually be stem squamates outside of the crown, giving us some sense of the origins of the group. And so you have this vast data set, which comes with its problems. And those problems are really intriguing. And this is going to branch us into some of the other questions that you've got for us. But this is part of what's really motivating about the group. Whereas, as Tiago was saying, as a paleontologist, you can work on some groups which are completely extinct. So you have none of the challenges of the crown, say, if you want to work on sauropterygians. If you're interested in plesiosaurs or stem sauropterygians, basal groups, placodonts, it doesn't really matter. You have none of that. Whereas with Squamata, we have the whole picture. And then we have what appears to be right now the irreconcilable interrelationship stories told by molecules as opposed to morphology, let alone what happens when we blend the data sets. So there are lots of really spectacular problems and great challenges with the group.
Thank you for your answer, Mike. So what he basically just said for the audience is that what's really cool about squamates is not just the fact that they have such great diversity and it's all undescribed, but also that there have been these longstanding mysteries about their evolution. And in addition to this, that the fossil record that they contain is really fascinating. So thank you for that answer. It's a really great one. And I think that David, you probably have something to say about more about the living diversity with your research. So I was wondering if you'd mind also answering.
Yeah, so for someone brand new to paleontology, I guess I'll talk about my introduction to squamates. I started off interested in reptiles, and that came from like a childhood fascination. I was really interested in anatomy. I just thought they looked so cool, and I was interested in paleontology. So I started off working on some descriptive work on the cranial or the skull descriptions of some extant lizards, and that research kind of got me more attuned to how much we know about diversity of osteological variation in modern lizards. And so that has been a real focus of my research, and a real passion of mine is to increase our understanding of morphological variation in living lizards. And like Tiago was saying, there's a lot of unknown diversity, and there's a whole lot of species. So there's a whole lot we don't know. And I think one thing that's very exciting for me is to learn more about this and how we can use increased knowledge of that variation in the modern lizards and apply it to understanding the fossil record. And then, like Mike was saying, lizards have a fossil record, but also they're an interesting group, or squamates are an interesting group because they also have all these modern day representatives. And so part of my research is tying in the fossil record with the modern day species. And I try to understand how we can use the fossil record to understand responses to environmental changes. And I think and then tie that into understanding how that can inform us for modern day species and populations to try to understand how those modern day populations might respond to similar dramatic environmental changes. So I think with squamates, it's a very very diverse group, a lot of extant modern representatives, and a lot of different avenues for research based on both anatomy, phylogenetics, and also ecology and evolutionary questions.
Yeah, thank you for your answer, David. So what's kind of cool about your answer is that in some ways it sits between both Mike's and Tiago's, because you study morphological variation. And as Mike was bringing up, we really only have morphological variation in the fossil record, which can often come at odds with evidence from molecular evidence. But then at the same time, because we have this diversity that Tiago was talking about, like you said, David, we are able to really study that variation a lot better using these living groups. So thank you so much. This was also a very great answer. And that brings us to our final panelist, Amelia. And I just wanted to preface this because Mike already foreshadowed it. Amelia does work with mosasaurs, which are also squamates. They are fossil squamate and they're aquatic. So take it away, Amelia.
Yeah, so a lot of the main reasons that squamates are important have been covered by everybody else, but I'd like to highlight two of the reasons that I chose squamates, and more specifically mosasaurs. But in general, if you want to work on a fossil group, sample size is an issue, and it's much less of an issue with squamates than it is with some other groups, like certain dinosaurs. So that makes it actually possible to do certain types of statistical tests on them that you can't necessarily do on an animal that's represented by, say, a single individual. And then, yeah, like other people have said, they're a really diverse group. They've basically done everything ecologically except powered flight, as far as we know. So basically, if there's a question in biology that you're interested in, there's a squamate that you can study to solve it. And oh boy, now I gotta remember the last point that I had. Oh no. Yeah, so yeah, there's a lot of questions that are left, and oh my goodness, I'm completely blanking. There was another thing that's cool about them that I'm not remembering, but
Everything is
right.
cool, no worries.
They're all cool. I remembered it. The other neat thing about squamates relative to a lot of extinct groups is that morphologically they are still, even the fossil ones are relatively similar to ones that are alive today. And so it's much easier to, even though we can't actually study their DNA or their embryos, we can do studies like that in extant squamates and get a pretty good sense of what was most likely happening in the extinct groups. And there are a lot of labs that are doing a lot of really fun, cool work with, for example, lizard embryos. I did, for my undergrad research, I was part of a gecko embryo lab, and I know that there's at least one or two, if not more, labs that are focused on embryology in anoles. So using them as model organisms to solve big picture questions.
Yeah, can I just quickly add to that, because one of the interesting aspects is that differently from what we see with other vertebrate groups like tetrapods, especially in the case of mammals and birds, the major groups of squamates, they are much older comparatively. So we have no questions that they originated prior to the Permian, to the Cretaceous Paleogene boundary, whereas for birds and mammals usually have some conflict in terms of the origin of the modern families. Before or after that, from conflicts between the molecular clocks and the fossil record. For squamates, there is no doubt a major group actually originated substantially before that. They were quite well established by the end of the Cretaceous. So there are much deeper time kind of problems associated to understanding squamate origins and evolution.
Yeah, and I'll add, and that's talking about the crown group.
Yeah.
And I will say too, just to follow up on a comment of David's and discussion of the crown, there are so many known species of living squamates for which we have no published descriptive work, osteologically, morphologically. I mean, I was surprised a couple of years ago when one of my students and I went to publish on what we'd observed about the iconic Galapagos marine iguana. anatomically speaking, and there was nothing in the literature describing the cranial anatomy of that icon of modern Galapagos critters. There are so many things we don't know even about the extant group, and there are 11,000 species to choose from. It's a huge untouched data set.
Yeah, thank you all so much for your answers. This has been great and really interesting. So we have our next question, which is, and I think that this ties a little bit with what you all were just saying right now, which is, what are some challenges about studying squamates unique to them, which might not come up as often in other groups? And I think this in a lot of ways relates to what we were talking about, because, you know, squamates are so speciose and there's so much variability in them, as we were just talking about. And, you know, that's often very difficult and even, dare I say, overwhelming for researchers to study. So I was wondering, I guess, Tiago, you could start off by your answer to that question.
I think there are several challenges, but I think I would like to pick two challenges from very different perspectives. One is from the perspective of the fossil record and anatomical data. Well, there is some of the challenges Mike posed, which is extant knowledge of the extant anatomy, but we also have a huge biased sampling of the squamate fossil record. Well, we have a biased sample of everything, but with squamates, it's quite crazy. We have, among non-ophidian squamates, because with snakes you get a little bit of a different story, across the entire Mesozoic, we have about 200 species described for the Northern Hemisphere, so the global North in general, and most of the continents that were once part of Laurasia. Whereas for Gondwana, we have about 12. So that's for about, from the early Triassic to the end of the Cretaceous, which is a pretty long period of time for such a long biased record. And things have been changing more recently with increased interest and knowledge of squamates from the global south, but we still have a huge challenge in kind of compensating for this historical gap in terms of research effort in understanding squamate paleontology and squamate fossils from the global south. And the second part is from a molecular perspective, which is the fact that we're still lagging behind in terms of our understanding of squamate genome evolution compared to birds and mammals, as like they're twice, as we mentioned, twice as diverse as mammals. They are equally diverse as birds, and yet we are substantially lagging behind our understanding of and sampling of genomic diversity for squamates compared to what has been developed for other groups. And with that, it hampers our understanding of squamate relationships, even with the rich amount of data we can obtain potentially from the molecular record, including relationships within the iguanian families, for which we still have a lot of open questions, and a few other areas within squamate systematics. And so it may sound that sometimes from an outside observer that squamate systematics from a molecular perspective is quite stable and there is a very straightforward answer, but in fact it's not really the case. There is also a lot of internal inconsistencies within different molecular hypotheses, not as much as we have with the morphological ones, but still there is a lot of challenges ahead over there. So those are the two biggest challenges from my perspective.
Yeah, thank you so much, Tiago. That's a great answer. So for those of you in the audience, now you know that there's both a combination of sampling bias in the fossil record because no one's looking for the squamates enough. And there's also this genomic difficulty because people aren't studying the DNA of squamates as much as they should. So thank you. And next up, Mike, what would you say?
I'll turn my mic on, that way it'll actually help. Yeah, I'll just follow up on Tiago's point about, and the one I guess I sort of alluded to earlier too, which is the genome side of things. But, and Tiago's heard this argument before, so, he can turn off his sound system completely. He doesn't need to hear it again. And it's going to be an ironic comment where I've just been touting the spectacle that is the data set morphologically and genomically in this case with the Crown Group. The Crown Group is also, ironically, a problem because the data set is so complete that the Crown Group becomes bomb-proof to any disruption by the fossil record. And so we're consistently seeing difficulties in trying to recover relationships with fossil taxa that may not necessarily fit into the Crown because their data is incomplete compared to the Crown. This is one possibility. It's not necessarily a certainty. And maybe the extant Crown is really a monophyletic assemblage relative to everything we might find in the fossil record. But my gut instinct tells me that that's not the case. But we've got such a solid data set that trying to come up with character or recover character states that might break that Crown apart. And for instance, find amphisbaenians to in fact be space aliens that landed on the planet a long time ago and are not nested within the Crown in the way that we understand the Crown, but could be derived from another kind of Lepidosaurian group we don't know, or a Lepidosauromorphan lineage that we don't understand the same way. This is one of the problems that I find. And I don't see it as an impediment to research. I see it as presenting a series of challenges and really important research questions. Is the Crown the monophyletic monster that it is, or is it just impervious to being disrupted because there is so much data to support that thing, and 10 morphological features in some really funky fossil taxon will never be able to break that Crown apart, even if it should be. So it's one of the challenges, and a big one for me.
Yeah, thank you so much for that answer. That's also a really interesting point. For those in the audience that don't know, molecular sequence data will often have thousands upon thousands of data points, basically, whereas a morphological data set, it's usually, if we're lucky, about 1,000 characters. So what Mike's talking about is that a lot of times the genetic data can just swamp the morphological data when people are doing their analyses. And this is a really good point, that it could definitely cause a lot of challenges for people that are working with fossils, where there are a lot of extinct groups that we don't necessarily know the placement of, because then the modern groups are just totally biasing our sample, like you just said. So thank you for that answer. And if anything is an alien, it's definitely an amphisbaenian. So I really appreciated that. Next up,
I like
David.
the fact that Tiago agrees.
And Scolicophidians, don't forget them.
Oh yeah, them too.
All right, some challenges for me. For me personally, one challenge, I'll start with morphology and then I'll go to genetics. So for morphology, one challenge that I have with squamates that's not necessarily in other groups is that squamate cranial, or basically skeletal elements in the fossil record, are often come as disarticulated elements, which means that in the fossil record, oftentimes you get one little bone, and you work with that bone to identify what type of animal it is. So knowledge of osteological variation in modern taxa is very useful in that, but since you only have some isolated skeletal elements, that is a challenge that you don't always have with other groups. Like mammals, their skulls are mostly fused, so you get more of the skull intact in the fossil record often. Sometimes you also get isolated fragmented bits, but with lizards and snakes in particular, their skulls are really kinetic and they're not fused or held together by really solid ossified tissue. It's a lot of cartilage connecting the bones. So that's one challenge that I've faced. And another challenge is that they're very small. So a lot of times when I'm looking through sediment for fossil squamates, I have to look under a magnifying glass or look very closely. Whereas for other things like dinosaurs, you don't have to look as closely to find those fossils. And so that's the morphological side of things for some challenges that I face. And then And on the genetic side, like Tiago was saying, there's not a lot of genomic data available for a lot of species. And part of my research uses ancient DNA, and that can be, and not having those reference genomes is a challenge, because if you're able to successfully extract that ancient DNA, and if you have no reference genome to map those extracted reads to, then you're not going to get as much data back as you potentially could. So I think developing more reference genomes for squamate reptiles is definitely a need, and it can benefit a lot of different aspects of research.
Yeah, thank you so much for your answer, David. What was really cool about it is that you have this element of the morphological methods that you're talking about, where you need to really work on squamates differently than most people. And then you also have with the genetics methods, especially with things like ancient DNA, where it's often, really fragmented and damaged. And, you need really good genetic resources like everyone's been talking about, but you don't have them to be able to properly study them and answer questions with them. So that's also a really cool perspective. Thank you so much for your answer. And next up, we have Amelia.
Right. So I'll agree with everything that's been said so far. On top of that, as I believe mentioned a couple times now, on the one hand, it's really cool that squamates are super diverse. On the other hand, it makes studying the fossil record of squamates a total nightmare. Because regardless of the relationships of all of the groups to each other, it's clear that convergent evolution is rampant in these things. And without the DNA, it's really hard to have a good idea or a good sense of what is truly convergence versus homology and all that. So it makes things tricky. It makes for some fun academic debates. But, regardless, we're hoping to get a better sense of that as we go forward. And then another issue or problem, which is something that Mike mentioned earlier, which is that you'd think that these things being as common as they are would be better illustrated in the literature. And they're just not. So it is really difficult when you're first starting out to work on any group that's not like iguana or something that has been, a hallmark of comparative anatomy classes for decades. So yeah.
Yeah, thank you so much for that answer, Amelia. And I think what I'll probably do is reverse the order of the people I'm asking questions to now, so that everyone gets a chance to answer in a first or second order-ish. So, Amelia, what would you say the job of a squamate researcher is like, and how might it be similar or different to a paleontologist who studies other groups?
I would say that in my own experience, it's a much more chill time than other paleo groups. It seems like to kind of call back to there are a lot of disagreements within squamate researchers about the different relationships of groups. But that being said, I've felt like the community overall has been really supportive and helpful. And in general, less aggressive and hostile than some of the, dinosaur research groups, let's say. So yeah, no, I mean, it's been nice and welcoming in that regard. But then, as for the reason I mentioned earlier, was a little bit frustrating to trying to get started working on these animals, because there are fewer resources than you might expect for getting your feet off the ground and studying these things. Let's see. Similarity, though, I mean, especially if you're a fossil squamate researcher, we're still dealing with the incompleteness of the fossil record. Even if the sample size is better than dinosaurs, it's still never going to be complete. And we're dealing with all the same limitations that other fossil groups have as well.
Thank you so much, Amelia. That's a really great point. And it's also very heartening, since, for you to say that in your experience, people generally tend to be really nice when they're studying squamates, because, I think that if anything, we've all just discussed how people are constantly disagreeing on things, but it's nice that, you and I'm sure that other people in this room feel like there's still a cordial environment, which is something that, a lot of people might not say when they're working in other groups, which is really awesome. So thank you so much for your answer. And then next up, David.
Yeah. So what is the job like? For me, a lot of time is spent looking at, small fossils through a microscope. I do a lot of stuff with CT data too. So on my computer, I can look at 3D models of skulls of lizards and snakes. And then that's most of my time. And then part of my time is in the lab, the ancient DNA lab. And with squamate fossils, since they're so small, it can be a little bit more challenging than if you're working on, for instance, like a mammoth tooth. A small little lizard bone is a little bit harder to work with. But I think, like Amelia said, it's kind of a small world in squamate paleontology, and everybody's very collaborative and friendly, I've found so far. So I think it's a fun job.
Thank you so much for your answer. Mike, I see that your mic is off. Do you want to go next?
It's on. You want me to tackle this one? What's the job of a squamate worker like? And, to tackle a little bit Amelia and David's comments about a congenial and cordial academic debate environment. I don't know, my own personal experience might counter that slightly. But in any case, what's the job like? Well, you can be a squamate evolutionary biologist and vertebrate paleontologist in a number of ways. Let's just tackle a couple of the more mechanical or maybe even mundane sides of it. Curators at museums, professors at universities. These are two of I guess the classic ways to practice the science. And in that particular case, the career of a university professor, a museum curator, is a brilliant way to go about practicing science and making a living at the same time. There's absolutely nothing I can say bad about it. My entire academic career, I've had nothing but the greatest opportunity in working with students and watching them progress. I don't want to get all teary-eyed, but I'm proud as I could possibly be of Tiago's most recent success. And I've got a number of students who've been fortunate enough through their skill set to do exactly that. So it's both being a squamate worker and a professor, working with young people and learners, I guess is the better way to put it. It's not just young people in university environments, whether they're undergraduates or postdocs, or even better yet, collaborating with the people who are now your colleagues that were once your students. But that's sort of big picture stuff. Squamate work and the squamate paleontology or evolutionary biology community. On the paleo side, it's small. There aren't very many of us. Current professorial or curatorial appointments on mosasaurs alone, as Amelia knows firsthand, there aren't very many of us on the entire planet. I'm really the only mosasaur specialist with a PhD in my back pocket, working in a North American museum or university. Natalie Bardet's in Paris. She's trained a few students in the subject area. And then there are a couple of other spots, but not necessarily populated by very many of us at all. On the snake side, there are 3 or 4 of us on one side of the snake battle and 3 or 4 on the other. It's a pretty small community. There are more herpetologists practicing as evolutionary biologists. But again, as Amelia knows, most of them don't even think of mosasaurs as squamates. There's one of our great challenges out there. When they talk about lizard evolution, they never mention mosasaurs, which is weird as weird could be weird. But in any case, I think the job's great. We've chatted about the data set. It's vast and the problems are complex. I wouldn't study any other group now. I even like the fact that we've got such major morphological transformations in the group, from limblessness in multiple groups and elongation to some of the smallest frigging living terrestrial tetrapods out there. some of those tiny chameleons and itty bitty little anoles, all the way up to the fossil record of, mosasaurs, for heaven's sakes, some of the largest bodies we know of outside of modern cetaceans. So it's pretty cool. The job's great.
Yeah, thank you so much for providing some foreshadowing for people that really want to pursue squamates and fossil squamates in particular as a career. So thank you so much. And next up we have Tiago.
Thanks, Megan. Yeah, that's absolutely true. We have like a much smaller number of paleontologists working squamates compared to dinosaurs or mammals. Well, everything is small compared to dinosaurs or mammals in paleontology, but at the same time, even I would say even among herpetologists, there are not as many as we have as with people studying mammals or birds. So it's also not as big as. And I find that in the everyday consequence of this, the practical consequences, that we actually have a lot of interesting questions that are still untackled, and it just opens up a lot of opportunity of research, because there is a lot of big questions which are really have been tackled very few times or none or nothing at all, and it's just an amazing opportunity. That's one of the things that really attracted me to squamate research to begin with, because there is a lot of very interesting questions that have been rarely addressed. So it's a lot of opportunity. So being like a small group is not bad. It's actually can be good sometimes.
Yeah, thank you all so much for your answers. These have been great. And I was wondering if the audience members had any questions before I asked the last two, just to make sure that if anyone in the audience had some questions that they really wanted to ask the experts, that they get a chance to do that first. So if any of you in the audience have questions that you wanted the panelists to answer, do you can just put them in the chat or start thinking about them.
Well, in the meantime, if I may, Megan, you could describe a little bit of your own research, what you're up to.
Oh, sure. So I'm a first-year PhD student. I typically study neurosensory evolution using geometric morphometrics. But like David, I also do work in paleogenomics, which is the study of ancient DNA. And I also work in museomics, which is the study of historical DNA. And the taxonomic group that I specifically focus on right now is chameleons. So I'm studying their evolution. And I'm using a combination of the historical DNA, genomics, ancient DNA, and morphology in order to resolve the systematics, so the evolutionary relationships of that group. And it seems like there actually is a question in the chat. So I'll ask this to Amelia first. How do squamate researchers tackle a lot of the convergent evolution in squamates?
Sorry. I'm gonna defer to Mike and Tiago on this one.
You don't get off that easily. Yeah, moderator
Right.
asked you first.
All right. I'll answer how I typically deal with this. So with working on mosasaurs, I have to deal with this quite a bit. And basically, my philosophy about this is you do what you can with the bones, right? So because we don't have the DNA, we are 100%, at the mercy of what the skeleton can tell us and our own interpretations of what those shapes mean, and how they might fit together. So honestly, and this is something I think I can't remember exactly who said it. It's kind of an issue among modern or extant herpetologists that I feel like they don't really take the fossil record into account perhaps as much as they should. Because really we need to look at both the things that we understand based on DNA and the things that, don't have DNA, like the fossil record, to actually get the complete picture of what exactly is going on. Sorry, hello. Yeah, I guess so. My own philosophy with it is we have to take everything into account to the best of our ability, and this involves, looking at the relevant groups for whatever question you want to work on. So for something with snake origins and that name that I'm too tired to pronounce right now. Yeah, just doing your due diligence and using all available data to come to an answer with that.
Yeah, thank you. Sorry to put you on the spot a bit
It's
there,
okay.
Amelia. So
Spicy question right out of the gate. Goodness.
yeah, do you mind clarifying? So with mosasaurs, where does convergence usually show up for you?
So the two aspects of mosasaur, or I guess three aspects of mosasaurs that make them difficult to. So for background for people who don't, who are not in the lizard weeds as the rest of us are, mosasaurs have been historically difficult to place within Squamata, with two major camps coming out of this. So there's a group of people who believe that they're more closely affiliated with varanoid lizards, and another group who believe that they're more closely affiliated with snakes. And the trouble with answering that question is that mosasaurs have done a couple of things. They've elongated their bodies, they've reduced their limbs, and they've evolved these really flexible skulls, more or less. And those are all things that snakes have done as well, which could make it appear, or make convergence appear to be homology, or vice versa. And again, without a let me phrase this carefully. Without, the entire fossil record, which we will never have, it's really hard to have the complete picture when we're missing so many pieces. And I defer to Tiago and Mike because they've done a lot of their work on this question specifically. Most of my work is focused on in-group relationships of mosasaurs to each other, so that I don't have to worry about that for now as a PhD student trying to learn a lot all at once. But it's really fun, evolution, like scientifically. It's really interesting that such a well-known group like mosasaurs is still a little, cloudy to us where they actually go. And I think part of this is because, as others have said, like, there's such a small group of people working on fossil squamates, much less mosasaurs, that, there's still a lot of work to be done. And there's always, sorry, I was being threatened. There's always, with evolving technology, we're going to have more and more resources to actually address this problem, like CT scanning and finding new localities and all that other stuff.
Yeah, thank you so much for your answer, Amelia. This is really cool. So I guess next I'll ask David, do you have experience with convergence, and how do you approach it?
Yeah, I haven't had probably as much experience as some of the other panelists here, but with convergence, and specifically in terms of phylogenetics, I think it's important to think about how you're constructing your character matrix. Basically, how you're constructing your data matrix for trying to understand these evolutionary relationships, cognizant that convergence is, a phenomenon that happens. And so that might be including or weighting characters differently. So I think it's important just to think about that a lot, and not just throw everything into one big character matrix and seeing what pops out. And that's what I'll say.
Yeah, thank you so much for that answer, because it makes specific reference to how people are actually approaching this question to begin with. And, when you're designing these character matrices, like you said, we're coming up with morphological characters that we hypothesize might be influencing the evolutionary relationships. And, obviously with convergence, it becomes much more difficult to come up with those hypotheses because things might be similar, even though they don't necessarily share a homologous trait. So yeah, thank you so much for your answer. Mike, did you have
Oh, sure. Yeah. I've got some thoughts on this one too.
How many hours do we have?
yeah. How many hours have we got for this? To follow David a little bit, and we'll put it in sort of methodological terms. Homoplazy, which I'll synonymize for this few minutes we have with convergence is more common than homology, more often than not. The question was about tetrapodophis, long and skinny, right? I mean, many things are long and skinny. Not all of them are squamates, yet there are common themes in the way that other vertebrates and other tetrapods become long and skinny. Even eels lose their fins, for heaven's sakes. So, right away, it's very clear that characterizing gross morphology in some sort of broad terms, like the issue with tetrapodophis as a snake or as a dolichosaur based on nothing but, elongate. That's going to miss where the homologs might lie with finer scrutiny when you're constructing your characters and scoring your states. That would be, just the tip of the iceberg. How many groups of, and now I'll stick with squamates and I'll avoid aistopods and all sorts of carboniferous early tetrapods. How many squamates are limbless and do so convergently, independently, or in terms of your data set, homoplastically within Squamata? I mean, there are a lot. Dozens of them are completely limbless. Just the genus Larista among Australian skinks, there are about 140 described species, and they show every version of limb loss and limb reduction from 5 toes on each foot and little hand down to completely limbless, or one's got some sort of humeral peg and 2 toes on the back limb, and I mean everything in between, right? So clearly convergence is rampant in our data set, which means that homoplasies are going to pop up more frequently than you can imagine. How to weed that out? Careful character construction and a solid notion of how it is that you're constructing characters and what your concept of homologies really is. What is that foundational primary homolog concept that you depend on? You've got to have one. And, thinking of Amelia's work with mosasaurs and my work with mosasaurs, right now, for the people out there who might be listening who aren't aware of it, there's a big discussion about homoplastic or convergent evolution simply in the paddles of mosasaurs. If current phylogenetic schemes are accurate, then it looks like it probably happened at least 3 times, which means even the little flippers in a mosasaur are not commonly sourced from one single ancestor, but evolved at least 3 times. That kind of stuff is fascinating because it changes as well the way that we see the process of evolution, not just the pattern. We need that pattern first before we can hypothesize process, but there you go. Now you have a triphyly of the mosasaur paddle. That's totally cool if that's what happened. But again, it all depends fundamentally on how we construct our characters and the way we conceptualize our homologs. Yeah, it's good stuff. So yes, study legless lizards. Lean right into it. I see that in the chat.
Yeah, just to complement that, of course, if you work on groups with external representatives, the molecular data helps to split those apart quite easily as well. So if it was not by the molecular data, probably the predominant hypothesis nowadays would still be of like all limbless groups together. And I mean, not all of them, but many of them. And that helps a lot. Yeah, or something of the sort. So like that definitely helps. Whereas if you have an entirely fossil group, those debates can continue forever because sometimes the convergence is just too strong to discriminate what's actually representative of the same historical process of evolution or separate ones converging to the same final phenotype. But aside from that, with the challenges, because that's, a lot of challenges when it comes to systematics, which is, a lot of what we do. But at the same time, in terms of evolution biology and research questions in that field, it presents us with a lot of opportunities because we have at our disposal a unique study system with a crazy amount of convergence, way more than you have in some other groups. And by happening so frequently, we have every single stage into, let's say, limb development. in Larista, in gymnophthalmids, which are glass lizards from South America, we can see fully developed limbs, no limbs at all, and every single stage in between across different species, and sometimes within the same species. So they provide an excellent study system for developmental biology and genomics to understand the genetic underpinnings of these adaptive changes and the ecological basis for some of that as well. So there is and there are lots of labs, not lots, but there are some labs who are specifically dedicated to answer those questions, because it's very different from, let's say, in birds. We don't have lots of living intermediaries between flightless and fully flight capable birds. We have reversals to flightless birds, but we don't really have as many as intermediates in their condition and fully functionally capable as we have in squamates for limb loss. And I think one of the most interesting things that we have been seeing in recent years is the recognition, not just that happens with the extant fossils, but also in snake evolution. Mike and I were part of a paper a few years ago where we kind of realized by the collection of the available data across the past decades that along with our colleagues there, that the condition of partial limb development in snakes is not just like an ephemeral condition, a transitional stage where the let's say the most successful condition of limblessness was actually the most strongly selected for. In fact, snakes probably had partially developed hind limbs, but still quite well developed for probably 70 to 80 million years before they went fully limbless. And we see that with the extant record as well. We have fully successful clades, highly diverse ones with partially developed limbs. So that tells a lot about functional morphology there and how those intermediary forms can actually be strongly selected for, and they just don't need to be quick transitional forms into, let's say, an optimal phenotype into an adaptive landscape. So for understanding evolution processes, it's really a full plate of opportunities there.
Yeah, thank you so much, everyone, for answering that question, and I guess slightly being put on the spot by it. So we've come close to the 4 o'clock mark. So I think what I'll do is I'll ask you all to possibly wrap up with maybe 1 or 2 thoughts on what the biggest knowledge gaps and areas for growth in the field are. And then after that, it seems like there are a lot of questions in the chat. So I guess I'll stop recording after you all give those 2 statements. And then if anyone wants to answer those questions or might be able to be fielded them, maybe we can handle it by having people reply, or yeah. All right. Thank you so much. So yeah, Amelia, did you want to go first?
Sure. Lizards are great, and if you take away anything from this panel, it's that we need more people working on lizards, so that we can actually, start. I don't want to say actually, because we've been making headway on these questions that we've brought up several times now for decades, so that we can continue to make headway on these questions and, continue to implement new methods in studying squamate evolution, particularly in the fossil record. Yeah, no, that's, yeah, there are my 2 cents.
Agreed. David?
Yeah, so I think some big gaps right now for me that I see is understanding better the morphological variation extant lizards. And that's more than just, for me, just having knowledge of what each species looks like. That's looking at what is the morphological variation within a single species, what are the ontogenetic patterns that we might see in those species, sexual dimorphism as well, and applying all of those patterns of variation, that knowledge of patterns of variation, to the fossil record. I think having that knowledge in our arsenal for the modern will help us really be able to more confidently interpret the fossil record for squamates, which will help give us confidence in interpreting what we're seeing in the fossil record and trying to understand some of these questions.
Thank you. That's a great takeaway as well. What about you, Mike?
Wow. Do we have another hour so I can identify where I think we've got lots and lots of things worth investigating? You know what? I'll answer it, Meghan, by sort of big picture stuff, I guess, is how I'm going to tackle this. I'll just say that, you know, for me, there are more questions, a legion more questions than we currently have answers. I see there was a little question in there about venom delivery systems. I just recently published a paper with a couple of past students, Alessandro Palci and Aaron LeBlanc, on venom delivery systems and the presence of plicidentine in the dentitions in the teeth of snakes. Yet a classic synapomorphy for decades of serpentes is that they lacked plicidentine. And in fact, once we started looking for it, we found it everywhere. And it's implicated in the development of the venom groove because it's just infolded dentine. So what's the point of that story and my questions versus answers? It's that don't be afraid to, make sacred cows into burgers, topple dogma, wherever it sits. And it might be a character complex, or it could be missing information and multiple questions on an entire group. It's like my point earlier about the Crown Group being an impediment in its own way, just because of the vastness of the data set. Don't be afraid to kick that can. So ask your questions, get specific about characters and character states that are supposed to be the golden calf of synapomorphies that unite a group. Check it all out. Don't be afraid of any of it. There are millions of things to be done. That's where I'll leave it.
Always great to leave it on. We have so many questions. I like that point as well. Thank you so much. Tiago, what would you say?
Yeah, there is an infinite number of interesting topics that are and big gaps. I think we actually have covered some of them, geographical sample biases and genomes. But I think my last point would be in terms of the future of squamate research in academia and students, because we currently have knowledge of squamate anatomy is it's on a down path. It's kind of dying. It's so hard to find people who actually are currently being trained to know squamate anatomy, and also you can always use information from genomics, and there is a lot of people being trained in genomics, squamate genomics, but not so much into squamate anatomy. Outside of people interested in paleontology, you have very few people who actually are working on squamate anatomy, and we need to keep that alive in order to be able to challenge all of those questions, have a thorough understanding of squamate morphological evolution and fossil record and all of that. And without that knowledge, we will never be able to tackle the big changes in the phenotypes and changes in body plans and a good understanding of the fossil record and so on. So we definitely need more people investigating squamates and being fully trained on all aspects of organismal biology, the anatomy, the molecular side as well. So an integrative approach, but most especially, and I mean, it's not, I think here everybody must have an appreciation for the anatomical data, but this is definitely something that is not so widely appreciated when you talk to an audience focused on extant taxa. So that's definitely one of the take-home messages that I usually bring up to them.
Yeah, thank you so much. That's also a really great note to end on, especially because everyone here is, trying to add to that understanding of anatomy. And as we talked about, it's just so hard. So thank you so much for that. And now I'm going to stop recording, but if any of you all want to stay to answer some of the questions in the chat, that might be helpful. But you don't have to if you're busy. So yeah, thank you all so much for for this panel. This has been great. And I know I have learned a lot just by being here, and I'm sure that everyone else in the audience has learned a lot too. So thank you all so much for bringing your perspectives. This has been phenomenal.